We have created short videos to summarize a book entitled Learning the Brainstem. The voice you hear is AI-generated, as clear in diction as we can make it. LTB 14 continues our tour of medulla. In a routine MR scan, you get maybe 3 axial images of medulla in a study. I arrange them from higher to lower as we move from left to right across the screen. So, what is the value of examining many more axial photomicrographs of medulla that we have at our disposal? Simple answer. Because a lot happens between the image furthest to the left and the middle image. We will see what happens in this video. And similarly, a lot happens between the middle image and the one furthest to the right, as we will see when we complete our tour of the medulla in a later LTB video. Here are 5 axial sections of medulla, starting with the highest, almost rostral in the upper left-hand corner. Then, as the white arrows indicate, we pass to the most caudal of these 5 sections in the lowest image to the right. At no point do we visualize the nucleus of cranial nerve 12 because all the sections are above that nucleus. Let us take the lowest of the 5 images and enlarge it. We place a white line in the midline between the 2 pyramids. Dorsal to the pyramids, as indicated by the green shaded area, is white matter on either side of the midline. A medial lemniscus sits on either side of the midline. It might help you to visualize the 2 medial lemnisci as a headless person standing on the pyramids. The person is without a head, because sensory representation of the face in the lemniscal system happens in midpons at the level of the main sensory nucleus of CN5. Lemniscal information will pass from a main sensory nucleus of CN5 to the contralateral medial lemniscus in midpons. Also, paying attention to the midline, the person's right hemibody sense of vibration and light touch, for example, is located in the left medial lemniscus, and the person's left hemibody sensation to those modalities is located in the right medial lemniscus. We include the legs and arms because leg representation is ventral to arm representation. The orientation of a medial lemniscus will change as we ascend in the brainstem. The left image is medulla, where the left medial lemniscus is parallel to the midline. In the right image, in low pons, the medial lemniscus is perpendicular to the midline. In both cases, however, there is still somatotopic organization. In the medulla, arm sensory representation is dorsal to that of the leg. In the pons, arm sensory representation is medial to that of the leg. If we mention the lemniscal system, then we should ask where the anterolateral system, mediating pain and temperature, might be. The green shade covers right medial lemniscus, which transmits lemniscal information from left hemibody. The red shade covers the area of the ascending right anterolateral system, which transmits pain and temperature information also from the left hemibody. The 2 tracts head to the same place eventually, to right thalamus in diencephalon, but they are spatially discrete in the hemimedulla. We mentioned in LTB 13 that fibers arising from principal inferior olivary nucleus on one side will pass to the contralateral inferior cerebellar peduncle directly across the medulla. I indicate the left inferior cerebellar peduncle with a large grey circle. So, the territory that we mark in green includes not only lemniscal fibers, but also olive to contralateral inferior cerebellar peduncle fibers that run orthogonal to ascending fibers of either medial lemniscus. Moving dorsally, the yellow shaded area includes structures that we have encountered previously. The white dot sits in the area of the right tectospinal tract. Dorsal to it, as indicated by the red arrow, is the right MLF. Dorsal to the MLF on either side of the midline is a nucleus structure which, to be absolutely clear, is not the CN12 nucleus, because we are above that level. The blue arrow points instead to the right nucleus propositus hypoglossi. Propositus refers to something above, in this case above the level of the hypoglossal nucleus. The nucleus propositus hypoglossi is actually a longitudinally extensive column of grey matter that we can visualize in multiple sections. Here we indicate the location of the right nucleus propositus hypoglossi at multiple levels. We use this slide at the start of this video And, as a reminder, we pass from high to low across the images. The nucleus prepositus hypoglossy contains interneurons involved in the control of all eye movements. Its interneurons connect to and from many nodes. Among them, vestibular nuclei, paramedian pontine reticular formation, inferior olive, flocculus, nodulus, superior colliculus in the midbrain tectum, as well as cranial nerve nuclei 3, 4, and 6. Although the nucleus is involved in eye movements, its incoming and outgoing fibers probably do not pass through MLF. There is one additional thing to note in relation to the nucleus prepositus hypoglossy. Lateral to it, multiple levels, in white shading in all these images, is the vestibular triangle, of which we caught a glimpse even as high as pons. In this lowest image of LTB 14, the right vestibular triangle abuts the right inferior cerebellar peduncle. Draped over the peduncle is grey matter that we will address in LTB 15. Pause for a breath, because while we are nearly done with the LTB series as a whole, the medulla has not quite yet revealed all its beauty. As always, thanks for watching.